I think, as everyone is probably aware, the last 2 decades have given us amazing leaps and bounds in how we can actually look directly at the genetic sequences of all the species on the planet. And as a result, and particularly for agriculturally important species such as Atlantic salmon, we've developed a massive amount of new knowledge on the genomic variation. And for Atlantic salmon, this has given us tools such as information about what specific genes are underlying life history traits that are important in wild salmon. It's given us laboratory and bioinformatic tools that we can use to very quickly assess genetic variation in a large number of individuals in a very short timescale for a relatively small amount of money. And what this means is that we can use all this genetic information to design genetic tools that can be used cheaply and simply to address questions that are really important in the management and conservation of wild Atlantic salmonids. We're doing this at UHI Inverness. Now, I didn't come up with this acronym, but I quite like it. Our ASIG program uses genetic tools, and we work directly with management agencies and landowners who are managing Atlantic salmon stocks in Scotland. And we work with them to use our genetic tools to answer their management questions. So as you know, there are a lot of important questions when you're managing wild Atlantic salmon. We've had a few people talk about some of them today. For example, yesterday you heard about the decline in early migrating salmon. People are also very interested to know how many wild breeders are actually returning to their populations. Yesterday, you also heard about the problem of wild fish hybridizing with aquaculture fish. But today, I'm going to talk specifically about how we're using genetic tools to monitor the outcome of supplementary and conservation stocking of Atlantic salmon in Scotland. Now, thanks to Ian's great talk earlier, I don't really need to go through this slide because Ian gave you a particularly nice overview of the problems that can be associated with using stocking to try and restore and supplement Atlantic salmon populations. It's been very popular in the past, but it's very much out of favor at the moment because it has become obvious how many problems there can be with stocking. But with a continuing decline of salmon in Scotland and elsewhere, as you've seen in this meeting, it is becoming reconsidered as an important management and restoration tool. And the most important thing, of course, is that any sort of stocking program for salmon needs to be carefully planned and monitored. And one thing you need to do is to have a way of discriminating your fish that you've stocked from the fish that are in the population due to wild spawning. Now, in any situation where you're breeding offspring in captivity and then releasing them in the wild, you need a method to discriminate the wild from the released offspring. Now, one way you could do it is by putting a physical tag on your released offspring. In salmon, this would be a pit tag, or you might cut off its adipose fin. But of course, you can't do that, for example, if you're stocking out salmon in really early life histories. So an alternative way of doing it is to take a genetic sample from all the broodstock parents and then compare it to the genetics of juvenile or adult salmon that you're later sampling from the area where you've stocked. And you can discriminate your stocked individuals from your wild-spawned individuals because you'll be able to, using a equivalent of a paternity analysis, you'll be able to link the genetic signature of the juvenile fish to the genetic signature of the parents. So here's an example of a project that we're working on. We're working on this with the Tay District Salmon Fisheries Board. The Highlands of Scotland, the salmon rivers have been greatly impacted by hydropower development, which took place in the 1950s. And here's an example of the River Garry in the Tay catchment. You'll see from the postcard on the bottom left, you can't see the title, but it says Salmon Leaping on the Garry at Struie. So it used to be quite a well-known salmon river. But in the 1950s, they developed a big hydropower system around this catchment. And what they did is they pretty much removed all the water from the Garry River. And they also put in a migration barrier there so that the Atlantic salmon would no longer go up and spawn. However, in the 2010s, the hydropower operator SSE came to an agreement with which they would re-water the river and also remove the migration barrier. So now you've got, again, restored habitat where Atlantic salmon can spawn. But the question was, it was an open question whether wild salmon would actually go up to the newly restored habitat and spawn. So what the management board decided to do is they decided to kickstart things by doing a supplementary stocking program. So here's a map of the Garry. The migration barrier was originally down there on the bottom right of the system. And what happens is that the Fisheries Management Board, it goes to tributaries downstream of this map, so down that way. It collects returning adult spawners. For the first year, they are stripped and the eggs are stocked where they would have been spawning, but then the kelts are reconditioned, and in following years, they are stripped and the eggs are used to stock the Garry. So here's the fish in the hatchery. They strip them, and then they stock them by planting eggs in artificial redds in these two locations in the Upper Garry. And then the following autumn, they systematically go through fixed sampling sites in this entire river, and they collect genetic samples from juveniles, fry and par. And then they bring them to us. We run our genetic marker panel. I'm not going to go into detail, but for those of you who are interested, this is our view of our methodology. And what we do is we collect genotypes for all the broodstock adults, all the sample juveniles, and then we reconstruct the pedigrees using a bioinformatic statistical method. So what we end up is something like this. So just to explain, this is a pedigree. The parents are along the top, and they're linked by lines to their offspring. And if all the offspring are in one vertical line, that means they're all the offspring of the same two parents. So two things immediately pop out here. You can see that we have a large amount of juveniles for which we can't identify parents within our broodstock sample, and that means these are wild-spawned individuals. So adult fish have been returning to this re-watered area, and they are spawning there. We'll also see that there's a lot more closely related individuals within the stocked juveniles, as you would expect, than in the wild-spawned juveniles. So you could expect that the wild spawning is generating is maintaining a lot more genetic diversity than the stocking. Because we know where all these individuals are collected, we can also make some nice inferences about the sort of competition that might be occurring between the stocked juveniles and the wild juveniles. So you can see that the stocked juveniles are actually moving quite far and quite quickly out of their stocking areas. So So this shows the locations of juveniles that were sampled a year after the first stocking, and you can see that stock juveniles and wild juveniles are occurring in the same locations, and both of them are surviving quite well to the par stage. Okay, I'm going to skip this slide, but just to let you know, we're also doing these projects, for example, on a restored hydropower area in the Ness catchment. And I'm going to go on to our third case study, which is a much longer-term stocking program where we're able to look right the way through and ask what is the outcome of stocking, do the stock juveniles return as adults? So this is the Carron River in the west coast of Scotland. It's not impacted by hydropower, but the geomorphology, the relief of the area means that there's a lot of spate flows when there's high rainfall, as you can see in these examples on the left, and these very often wash out the salmon redds. So this is a river in private ownership, and the owners of the river, their number of the returning adults became so low that they were concerned that the population was going to go extinct, so they instituted their own supplementary stocking program. So this was started quite a while ago in 1992, and the first stocking returns would have been in around 1998. And you can see since the stocking program was instituted, there's been a very great increase in the number of fish that are caught in the river, but it's really unknown whether the stocking program actually was partly or fully responsible for this resurgence in number of individuals. So we did a case study of one particular stocking year. This was 2015. So we took all the broodstock samples from all the broodstock, and there's 3 different broodstock types that they used here. And then we took the samples from all returning adults in 2017, 2018, 2019. And we also took a little look at a subset of the smolts that were going out in the 2 previous years. What we found, quite surprisingly, was that, I say fairly surprisingly, was that these juveniles that were stocked out were actually making a substantial proportion of the adult returns. So we can identify the fish that were both parents came from the 2014 broodstock crossing, but we also identified a number of fish that had one parent from those broodstocks, and we infer that these fish were fish that were stocked in different years, but using the same broodstock parents. And once again, because we reconstructed pedigrees, we are able to also look at a number of different elements of what's happening in this system. For example, we can see an example here where fish from a single family are returning as adults in 3 different years. So by doing these sort of studies, we can really understand how the supplementary stocking is contributing to the restoration or not of a wild salmon population. Now, there's a caveat here, because using these sort of genetic markers, we can also ask the question, what's the ancestry of the fish that are actually being stocked? And in this case, it turns out, not particularly surprisingly, because there's a lot of aquaculture on the Scottish west coast, is that these fish being stocked are not just pure West Coast Scottish fish, but they also have a certain amount of ancestry from Norwegian aquaculture fish. So it's interesting to ask the question as to what sort of impact this aquaculture material might be happening, and whether the stocking program is actually inadvertently increasing the amount of Norwegian genetic material that's in the river. I would just give a quick mention, as my time is running out, that the sort of genetic genotyping panels we use for these projects are really easily customizable. So one thing you can do is add markers that enable you to interrogate specific genes that we know are important in salmon life history, and these can be asked to be used to ask additional questions at the same time as we're just looking at general population structure and stocking outcomes. Okay, just going to say I obviously didn't do this alone. We had a number of great staff at UHI assisting with these projects. I especially want to say thanks to Dasha Svobodova, who pretty much is doing all our lab work at the moment, and of course many project partners and various funders. Thank you.
Thanks very much, Victoria.